Hi everyone, I'm Miquel, a fourth year PhD student at the Serrano Lab at the Center for Genomic Regulation, or CRG, in Barcelona. And today I will talk about our latest publication on how we can identify and rank potential cancer driver exons from a given potentially any given sample. So, as you probably know, cancer driver genes have long been studied as genes bearing cancer driver mutations. However, alterations in the DNA sequence are only one of many other ways that the activity of a pathway can be altered and drive cancer. And alternative splicing is one of these alternative ways in which we can alter genes activity and a pathway's activity. And basically, this is what we focused in my thesis. So most genes, human genes, are fragmented into introns and exons depending on the conditions and through the activity of splicing factors. The same gene can be expressed at the same mRNA levels, but have very exon inclusion patterns. And this can contribute to determining whether a cell is in a healthy or a cancerous state. And in the case of cancer, splicing is so important that we find, although scattered, some examples of exons that bear cancer driver properties, such that if we perturb them with splice switching oligonucleotides, we're able to modulate tumor progression. So, given this, we wondered, can we map somehow of all annotated exons which ones are potentially driving cancer, and can we use that in our favor to prioritize which exons one should target if we have to make drugs against cancer growth? So, to start, we checked on prior knowledge, and if we look at genes bearing known cancer driver exons from the literature, here in yellow, specifically that come from the AS Cancer Atlas, we see that there is a relatively small overlap with mutational cancer driver genes that come from the Cosmic database, and also with pan-essential genes, which are also expected to affect cancer hallmarks, like proliferation. So, considering this small overlap, we have to, and the fact that the cancer driver potential of different exons has not been explored, we can have two hypotheses. So, it could be possible that the same the same mutational cancer driver genes have also drive cancer through splicing, but it could also be possible that splicing drives cancer through a different set of genes that has not been yet identified, because we have mostly been paying attention to mutagenesis as a driver of cancer. So, to try to tackle this, we came up with a statistical approach to infer which exons are potentially cancer drivers. So, to identify them, we consider that different cancer cell lines express different isoforms of the same gene, but also respond differently to knocking down that same gene. So, we hypothesize that potential cancer driver exons are those exons whose inclusion across different cancer cell lines correlates with their own gene's dependency, that is, the knockdown effect of the gene. With this, a statistical association, we created a set of models that we call Spotter that allow us to infer not gene dependencies, but splicing dependencies. And according to the score, that whether positive or negative of the splicing dependencies, exons could be either oncoexons or tumor suppressor exons, borrowing the nomenclature from cancer driver genes. So, after doing this, we observed that and consider that most of the genes with potential cancer driver exons, 500 out of 800 of them, could not be categorized in any of the prior knowledge classes. So, we wondered what are they what are they essentially. So, if we look at their mutational rate, we see that the genes with potential cancer driver exons are less mutagenized than the rest, considering all mutation types, except in the case of intronic mutations, where we would expect some neutral mutagenesis. Then we also observed that if we look at the pathway level, these uncategorized genes with potential cancer driver exons are found in similar proportions in a specific manner in the same pathways as mutational cancer driver genes and genes with that we know that have cancer, known to have cancer driver exons. However, not they don't their pathway presence does not correlate with pan-essential genes, suggesting that the genes that we have found are highly cancer specific, but are found, and they are, although they are not known cancer driver genes from a mutational point of view, they do share or contribute to the similar pathways, which is a hypothesis that was published actually in 2020, I think, by the ARIAS lab. So this makes this gives us the idea that genes with potential cancer driver exons seem to contribute to cancer through splicing rather than mutagenesis, and this makes alternative splicing quite an orthogonal and complementary process to mutagenesis as a driver of cancer. Because we found these potential cancer driver exons through statistical models, models allow to make predictions, and therefore we should be able to simulate with these models how perturbing single exons leads to changes in cell proliferation. So to validate that, we took data from two independent sources where they had performed an exon-level CRISPR screen that consists in using CRISPR systems to delete single exons at a time and measuring how this affects overall cell proliferation. And surprisingly for us, we saw that the correlations are fairly good between our model predictions in the x-axis and observed changes in cell proliferation. So essentially, it seems like spotter models allow identifying which exons are more important for cell proliferation in a given sample. So not only finding which exons have cancer driver potential, but also making a ranking among them, such that if we have to choose an exon to make experimental validations on or to further develop a drug target on, we can decide whether we want an exon that is going to give a strong proliferative effect or a milder one. And given this predictive power, we thought, why not exploring across the TCGA which exons that hold cancer potential have also therapeutic potential. So we used Spotter to predict which exons are the most harmful ones, and at the same time try to find those that are more included in primary tumors, such that these potential treatment that we would just validate in vitro would be to promote the exclusion of the selected exons and evaluate whether this makes cells proliferate less. So, and these experimental analysis for the eight cases selected were performed by Ludovica Ciampi in collaboration with Sophie Bonal and Juan Valcárcel. So the results are shown here, and basically here you see on the x-axis the different targets highlighted, and then we have the controls, and in the y-axis you have the normalized cell proliferation upon the treatment of the corresponding splice switching oligonucleotide. And we see essentially the take-home message is that in most cases we observe the expected changes in cell proliferation. And we were quite surprised in that, because we were thinking that, okay, our algorithm may be useful for ranking, but we were not expecting that in all cases we more or less rank them as expected, which matched our validation using the CRISPR screens. So Spotter, it seems that is useful for ranking exons according to their importance for cell proliferation, and this accelerates drug development in the end, because I would like to highlight also, well, probably because Ludovica worked very fast, but we managed to go from identification to experimental validation in just 6 months. And basically, compared to other types of drugs, this is quite fast. So after seeing how these how these exons with cancer driver potential behave in our models, we wondered whether our models of splicing dependency could be used to find mechanistic drug-exon interactions. So what we did is we associated the predicted splicing dependencies using also a very simple linear model with drug sensitivity profiles. And here we expected to find two types of drug-exon interactions, either direct or indirect. In the direct case, it will be exons that are found in the drug target, and indirect would be exons that are found either in genes that act upstream or downstream with respect to the drug target. And this upstream or downstream could be a pathway or a protein-protein interaction network, which is what we validated in this case. So what we observed is that although these associations are statistical, they followed an interesting distribution. That is that if you map all these statistical drug exon associations, we observe that we find more associations closer to the drug target in the first part than as we move away of the drug target, meaning that these associations recapitulate the mechanistic mechanistic interactions. And also, I would like here we used StringDB as the protein-protein interaction where we perform this analysis. But also we have in the paper, we have other analyses that seem to validate this. And next, to just have an example and not just have a systems bio result, we studied the drug-exon interactions for nuclein 3A, which is a drug with a well-established mechanism of action that inhibits proliferation by blocking the interaction between MDM2 and TP53, which is a tumor suppressor gene, in a way that, so essentially MDM2 ubiquitinates TP53, promoting its degradation, and therefore TP53 is typically less inactive. When we add nuclein 3A, it blocks this interaction, and therefore we have more TP53 active, which leads to downregulation of cell proliferation. So what we found with these drug exon models is this exon that is not at the binding pocket but behind the binding pocket between MDM2 and TP53. We find that this exon stabilizes this interaction, and therefore this is why we believe we find the statistical interaction, because what happens, we think mechanistically, is that when the exon is included, TP53 and MDM2 strongly interact, and therefore we need more nuclein 3A to inhibit all the complexes, therefore making the cells more resistant than in the situation where the exon is excluded and we have less MDM2 TP53 complexes. And to finish, we explored whether, given that we have the drug exon interaction models that allow us to find mechanistic interactions, we thought that these, because we have a model with which we can do predictions, we thought that these predictions could also be used to predict whether given cancer sample before treatment, whether it would respond or not. And in this case, instead of looking at mutations, as many other algorithms do, we would be just looking at the transcriptomic information and interpret it through our very simple models. For this validation, we use data from 49 ovarian cancer patients, and we see that even though we are tackling the problem with a lot of simplicity, our predictions match more or less the expected outcomes of the treatments, such that patients that were sensitive to the drug have a lower predicted IC50 than patients that showed resistance to the drug, or on average. But this further highlights the relevance of our potential cancer driver exons that we identified in this study, because they are not only informative of cancer cell proliferation itself, but by fusing them and integrating them with other data sets, we could, in this case, use them to extract mechanistic information from drug sensitivity screens, which we think that is just the start of many more analyses and interesting models. So to conclude, what I would like to take home messages are that most genes that we identified that have potential cancer driver exons were not known previously to hold a cancer driver potential. We suggest that alternative splicing is a mechanism that complements mutagenesis in cancer and is in any way hijacked as well. Then, using our models to perform in silico RNA isoform screening, with it accelerated identifying functionally relevant exons, and we believe that this can lead to have therapeutic applications. And finally, integrating these splicing dependency models with drug sensitivity profiles, we saw that we could uncover some mechanistic drug exon interactions and predict treatment outcome. Because, as I repeated several times, we did everything with very simple models. We believe that in this study, what we are saying is that we created a baseline for the systematic discovery and prioritization of cancer driver exons. And therefore, we think that with the given technology and statistical approaches and the current advances that are going on, there is a lot of room for improvement. And hopefully we make this field more attractive, because we think that the splicing is so complementary to mutagenesis that it's likely also a good target for treatments. And before finishing, definitely I would like to acknowledge my lab, and because during all these 4 years they have been very helpful and funny, and you for your attention. And also I would like to know that I started a year ago this Twitter page called Splicing News. So if you also like to know more about splicing or to keep updated about splicing, follow it as well. Thank you. I'm happy to take any questions.